is Paul. Welcome to my poster. Non-typhoidal Salmonella is the second most prevalent cause of bacterial gastroenteritis in the Netherlands. The three most dominant serovars are Salmonella enteritidis, Typhimurium and monophasic Typhimurium. And what we did here is to evaluate routine One Health whole genome-based surveillance of non-typhoid Salmonella. Now, the methods are very complex, but in summary we used core genome MLST, and what that actually means is that we take all of our sequenced isolates and we cluster them into groups. We zoom into the clusters that contain both human isolates and those from the food production system, because those contain information about transmission of Salmonella from the environment to humans. Now, those clusters are visualized in these figures. For this first one, it is the one for enteritidis, and each of these lines contain a cluster with human and food production chain isolates. So for instance, this first one, you can see these purple groups, and they are mostly derived from poultry, which you can see in green. So in summary, we see that for every serovar, we can link the majority of food production chain isolates to humans. And specifically for enteritidis, we see that the majority of isolates are derived from poultry. Whereas for Typhimurium, we see that the majority are derived from cattle and swine in red and in yellow. And lastly, for monophasic Typhimurium, we see that they're also derived from cattle and swine. So that covers the first goal, creating an overview of the surveillance data. And the second goal is to evaluate the effectiveness of source-specific sampling, which is only covered in the manuscript, which is still in progress. So with that, thank you for your attention, and thanks to all the people collaborating on this project.